Hi Ninja Nerds, in this video we're going to talk about specifically the Phototransduction Cascade. So we're going to dig into the layers of the retina a little bit more, talk about those layers, and then we're going to talk about how we can turn light rays into electrical signals that basically produce vision. Alright, so let's go ahead and start here. So the first thing that we're going to talk about is the different layers of the retina. So what am I doing here? If you see here, you guys are probably like, wait, where the heck did this come from? Don't worry, all we did is if you guys watched the video on the structure of the eye, all we did is we're taking a little section right here. We're taking a little section right here from the retina and zooming in on it. Okay, so now we're actually zooming in on all the different components of the retina. All right, so let's go ahead and get started on this then. So first things first, the retina is actually made up of three main cell layers. Okay, so you see this outer layer out here? This outer layer out here, which is made up of these maroonish-like cells. This is the outer pigmented layer of the retina. So what is this right here? This is the outer pigmented layer of the retina. All right, so the outer pigmented layer of the retina, this is an important layer. Why? Because the outer pigmented layer consists of these little granules. See all these dots in there? All those dots are consisting of a specific type of molecule called melanin. You know, melanin is basically like a pigment. It helps to be able to prevent what happens whenever light rays, because you know whenever light is actually coming into the eye. This sometimes can be a little confusing for people. Let's come up to this little eye here for a second. Imagine here I have the light rays. Okay, so here's the light rays. These photons of light are actually moving in through the cornea. They'll go through the actual lens. And then they'll actually move through the layers of the retina. So they're moving through the layers of the retina. So if you think about it, the rays of light are actually coming this way, this way, this way, and this way, and then they're stimulating these photoreceptors, which we're going to talk about. But what happens is these photoreceptors, some of the endpoints here, this little like bumped little edges part of the actual photoreceptors, they're kind of embedded. The discs are embedded into these actual outer pigmented layer of the retina. And why is that important? Because these melanin granules are important for absorbing some of those light rays and preventing the scattering of light and preventing the actual reflection of that light so that it doesn't disturb the normal visual pathway. So that's one of the important functions of this melanin that's within the outer pigmented layer. Another thing, if you remember, we said that there's going to be blood vessels. You know, there's what's called from the choroid, you're going to have these actual choroidal blood vessels. So this whole thing right here is supposed to represent a blood vessel. So this is representing a blood vessel from the choroid. And what happens is this blood vessel gives off some of the substances that we need for the actual retina. So what happens is certain things are actually going to come out of this actual choroidal vascular supply. And from this, it'll go into these cells, these outer pigmented epithelial cells. These cells will process that substance and then push it out here into the retina to provide nutrition or oxygen or other different types of substances to the retina by diffusion, okay? So that's one function of it. So one function is to prevent the reflection of light, to absorb the light rays and prevent it from scattering. The other function is to provide nutrient supply to the actual nutrient, oxygen, other different types of vascular supply to the retina. And it's acting like a barrier. It's acting like a barrier to prevent, it controls what substances leave the blood and come into this actual tissue where the retina is. So it's acting as a barrier. It's also giving nutrition and oxygen and other different types of supply from the blood. And it's actually absorbing any light rays to prevent reflection of the light and to prevent the scattering of the light rays. All right, that should be good enough for that bad boy. All right. If you want to know some of the discs from these actual photoreceptors, some of them actually degrade. Well, they actually get released. Some of these old discs here from these actual photoreceptors, these outer pigmented layer of the retina, they can phagocytose that, okay? So if you want, they have four functions. Absorb any light rays and prevent it from being reflected and scattered. They provide blood vessel supply to this area by diffusion, nutrient diffusion here. And they control what leaves the blood comes into the retina area, and it phagocytoses any types of cellular debris from like the actual photoreceptors, like their discs, okay? All right, good enough. Then we get into the photoreceptor layer, okay? So these guys here, all these cells here are our photoreceptors, okay? So photoreceptors, there's actually two different types of photoreceptors. We're going to take a little bit of time to discuss this. So this is our actual photoreceptor photoreceptor layer. So the photoreceptors, there's two types. What are those two types of photoreceptors? There's rods, and then there's cones. And we should take a little bit of time to talk about this because this is one of the big things here. So photoreceptors are two types. One is rods, and the other one is of cones. We should be able to differentiate between these two. Now, what are the rods good for? Okay? The rod's specific function is for a specific type of vision. They call this type of vision here scotopic vision. Okay? Scotopic vision. Scotopic vision. So it's more for dim light or dark light or like fuzzy dim light, right? So dim light and dark light. So they're more for actual dim light, dark light, or another way of describing it is like lights in different shades of gray. So light that is found in different shades of gray. So light in different shades of gray. Okay, so that's the function of these rods. They play a role in what's called scotopic vision. So controlling, being able to see in dim light, dark light, or being able to see in different shades of gray. Now, one of the things is because they play a role in our vision at that type, so basically night vision. You can also think about scotopic vision as like night vision. So these are the photoreceptors that are functioning during the nighttime. But here's the difference here. When you think about these guys, if you guys have ever been in a dark room, can you really determine, like if I have like a certain structure, could you determine every single edge of that structure? Could you have very precise like edge detection of that structure? No. I might not be able to tell the color of that structure either. So at night, certain types of visual acuity is affected. So they lose their visual acuity and they lose the color at night, right? So this is not going to be like a very great vision for acuity. So it's not good for visual acuity. Okay, so they're going to have a hard time being able to see different types of shapes and edge detection and also color. So they'll have a decreased ability to see color, decrease what's called edge detection, and decrease in acuity of the vision, right? Okay, so a decrease also in the acuity of the vision, okay? So there's going to be a decrease in this color vision. They're going to have a decrease in being able to determine detect edges on some type of structure, and they might have a decrease in the acuity. Okay, so that's for the rods. Now, the reason why they have scotopic vision and they play a role within this dim light, this dark light, this fuzzy light, the different shades of gray, is because they consist of a specific type of pigment. They consist of a specific type of pigment, and this pigment is called rhodopsin. So it's called rhodopsin. And rhodopsin is actually made up of two things. One of the things is it's made up of what's called retinal. Okay, so it's made up of a substance called retinal. Okay, the other component is a protein called opsin. So opsin is a protein and retinal is a pigment, and we'll talk about why that's important. So rhodopsin is the pigment consisting of retinal, which is basically a derivative. It's a derivative from vitamin A. So vitamin A actually can get converted into retinal if you oxidize it. So if you remove off of it 2 protons. So if you get off 2 protons off of it, you can convert vitamin A into retinal. All right, so that's rhodopsin. But rhodopsin is very specific to the dark light or the dim light or that fuzzy light, right? That is one of the big differences about rods. Is they're for scotopic vision. Dim light, dark light, different shades of gray. They're not very good at being able to determine color or edge detection or acuity, right? And they consist of a pigment called rhodopsin, which is made up of retinal, which is a derivative from vitamin A, and a protein called opsin. Okay, that's good for right now. Cones. Cones are cool little buggers. These guys consist of, they're actually very important for what's called photopic vision. Photopic vision. So they play a role in photopic vision. These ones are very good for visual acuity and for edge detection and for color vision, but they're not very good at being able to see things in dim light and dark light and different shades of gray. That's why they're just basically kind of opposite in function. So they're for photopic vision. So these is good for color vision. Now, what do I mean? Okay, what does that mean, man? All right, don't worry, I got you. our cones, we have 3 different types of what's called these pigments. You know, there's actually, we have it down here. Let's write it right here. There's a specific type of pigment in cones. We're going to kind of bring it down, same kind of thing here. This pigment that's found in cones is actually called photopsin. Photopsin, which is kind of like, it's also referred to as iodopsin. But specifically, we're going to call it here photopsin. And the reason why is there's different types. There's photopsin 1, photopsin 2, and photopsin 3. Why am I mentioning this? Because in photopsin, what it allows for is this photopsin pigment can detect certain wavelengths of light. You're like, holy frick, he's bringing chemistry into this. Don't worry. All I'm trying to say here is that these photopsins can pick up different wavelengths of the visible spectrum to determine 3 main colors. So these photopsins are very, very sensitive to 3 specific wavelengths. One is the actual blue part of the visible spectrum. Okay? So they can pick up the color blue. Another one is red. Okay, so they can pick up red within the visible spectrum. And they can pick up green within the visible spectrum. If you guys know a little bit about your actual chemistry, though, you'll probably remember the term Roy G. Biv. Right, Roy G. Biv. And all this was trying to say is that as you're going this way, right, the energy is increasing. So energy is increasing. Frequency of this light is increasing. But the wavelength, as it goes this way, the wavelength, which is here like lambda, this is decreasing. So energy is increasing, frequency is increasing, but the actual wavelength is decreasing. So in other words, if we were to pick up these colors that we were talking about, blue is higher in energy, but it's actually lower in wavelength. Green is actually going to be a little bit higher in energy than the red, but it's actually going to be a little bit more in wavelength. And then the red is going to be the lowest in energy, but highest in wavelength. Okay, so if you were to kind of write it like this one is actually going to have the highest wavelength. This one will have the lowest wavelength. And this will kind of be right in the middle, so I'm not even going to put an arrow there. I'm going to say this is kind of within the middle. All right? That's the whole purpose, is that these photopsins can pick up different wavelengths of light. That's it. So they play a role in photopic vision, which is for color vision, and because they're very important during our bright light, they're very good for visual acuity. So they're good for bright light vision, and they're very good for like visual acuity, right? So they're very good for visual acuity. So they can have they're very good at being able to determine the shapes of certain things, right? So edge detection. So they're very good at that edge detection, being able to see very, very precise things. Okay, so they're very good at precise vision. All right, that should be able enough to explain that, right? Now, same thing, the pigment here, this photopsin, it is made up of different types of things here. It's gonna have like iodine in a part of it too, so there's actually gonna be a little bit of iodine in this photopsin, but it's also gonna be consisting of opsin. Now, here's the only thing that's a little bit similar between these two. The way that they're stimulated is the same, okay? So their transduction process, which we're gonna talk about here in a second, is the same. That's kind of really the only thing that is the same between these two. Okay, their transduction process is the same. They both respond to light the same way, which is really odd, okay? All right, let's go ahead and keep digging in here. So now that we know exactly what rods are good for, we know what cones are good for, let's go ahead and keep going into the layers, and then we're gonna discuss how this light is actually getting converted into electrical potentials. Okay, what's the next part here? So we know that we have the photoreceptor layer. You know you have these special little cells located in between the photoreceptors within a horizontal plane. So because they're actually located within a horizontal plane and they're orienting these cells in a horizontal plane, these guys are called horizontal cells, okay? They're called horizontal cells. And these cells are very interesting. We'll talk about them in a little bit later whenever we get into the transduction process, like the signaling process. They can secrete certain chemicals called GABA, gamma amino butyric acid. And the gamma-aminobutyric acid can actually inhibit these photoreceptors to basically control. It's very, very important for being able to our adjustments when we go from bright light to dark light or dark light to bright light. So they help to modulate that activity, the adaptation in it. We'll talk about that. Okay, so that's the horizontal cells. Then we have this brown cell layer. This brown cell layer is actually consisting of what's called bipolar cells. Bipolar cells. And these cells are not bipolar, they're not manic and depressive. It's because they have one dendrite extension and one axon extension. Okay, that's it. And these are very, very important also. Now, in between, kind of like a vertical arrangement because they control the vertical motion. These cells here are interacting and they're modulating the activity that's occurring between the bipolar cells and these green cells here called the ganglion cells. They modulate this activity through certain types of neurotransmitters. These suckers right here, these maroon cells, are called Amacrine cells. Amacrine cells. And they're located within this inner plexiform layer. We're not going to talk about the plexiform layers, maybe in future videos, right? For right now, we're going to talk about these layers very simply, okay? So now, amacrine cells are going to be kind of within that, what's called the inner plexiform layer. Then, you're going to have this last layer here. This last layer is consisting of what's called the ganglion cells. And these ganglion cells are very, very cool. They have a dendrite extension that's connecting with the bipolar cells, and their axons are coming together and making one big old structure here called the optic nerve and form the optic nerve. Okay, so they all come together and form what's called the optic nerve, which is known as cranial nerve 2. Okay, so that's what we know so far. We know the layers. We know that there's the outer pigmented epithelial layer, the photoreceptors, which are made up of the rods and the cones. We know what they do. We know that there's the bipolar cells, and they're basically acting as the relay between the photoreceptors and the ganglion cells. We know in between the photoreceptors are horizontal cells, which are modulating their activity somehow. Amacrine cells, which are also modulating the activity between the bipolar and the ganglion. And we know that the axons of the ganglion cells are helping to form the optic nerve. Okay, so far we've done pretty good and we've just diced out the layers of the retina. We've identified what the rods and the cones do. Now what I want to do is I want to talk about exactly how this light ray, let's make this light ray a different color that would be more, it would make more sense. Let's do it in orange. So let's say that this is our light ray here. So this orange here is the light ray, okay? Here's the light ray. And what happens is I want to see exactly how this light ray, these photons, are producing these chemical changes and then electrical changes within these photoreceptors. Okay? So we're going to treat this structure over here as if it's a photoreceptor, meaning it could be a rod or a cone. We're going to specifically look at it as rods, but remember, the cone mechanism is the exact same. All right, so let's go ahead and dig in here. All right, first thing, what do we say? There's going to be these photons of light, right? So these photons. So here's these light rays. How does this affect this rod here? So we're looking at a rod right now, okay? We're looking at a rod. But again, the mechanism would be very, very similar for the cones. They just involve different types of pigments. So these light rays, they come in and they hit a special structure here. So here's going to be the outer segment. And there's going to be these little discs, right? Here's a little disc. And here's the space inside of the disc. Here's the outer membrane of that disc. What happens is these light rays will move through this outer segment and it'll hit a specific structure here on this disc. Here on this disc, let's do this in maroon, there's going to be that rhodopsin molecule. Here is your rhodopsin. So here's a rhodopsin molecule. And what did I say happens when it hits the rhodopsin? Well, first off, what's rhodopsin made up of? It's made up of retinal and it's made up of opsin. So let me show you something here. I'm going to show you the two components here. First thing I'm going to show you is the retinal. There's going to be this retinal structure here. And the important thing of retinal is it exists in two different forms, right? One is it's actually existing in what's called the trans form and the cis form. So let me just number this out real quick. 4, 5, 6, 7, 8, and then 9 here. Okay, so one thing that happens here is, here's our retinal, but there's a methyl group here on 3, on that third carbon there, and then there's a methyl group here on 7. But here's what happens. Let's say that this methyl group is pointing down, all right? Let's say that it's pointing down like this. If it's pointing down like this is going to exist in what's called the 11-cis retinal. If you hit this 11-cis retinal, that's what you're going to be doing. You're hitting with these light rays. If you hit it with this light rays, what it does is it converts the 11-cis retinal into a different structure. So here's the light ray. So this is the light. It's hitting the 11-cis retinal and converting the 11-cis retinal. It's just kind of flipping the structure around and converting it. I know it's so weird that that's all it does, but all it does is it kind of just flips the structure around a little bit here and converts it from this 11-cis retinal into what's called all-trans retinal. Okay? So then it's going to convert into what's called all-trans retinal. So now we went from 11-cis to what's called all-trans retinal. Okay? So that's what you did. You took the light rays hits the 11-cis retinal and converts it into all-trans retinal. Why is that important? Because then out of this, you're going to separate from the all-trans retinal the opsin protein. And then the opsin is going to go off and perform a specific function. Okay, so what happened here? We converted 11-cis into all-trans retinal. And then what happened here? From this broke away the protein. What was this protein that broke off? The opsin. What happens is the opsin goes off and activates a specific protein here. It activates a very, very specific protein. You know there's a protein over here. There's like a transducin protein. Let's say here's actually going to be this transducin protein. So here's a little transducin protein. So it activates a protein called transducin. This transducin protein will then go and activate a special enzyme over here. There's an enzyme here, and this enzyme is called phosphodiesterase. So it activates a special enzyme called phosphodiesterase. Why is this important? Let me draw another phosphodiesterase over here too. Here's why this phosphodiesterase is important. Because there's actually something else happening at the same time this is occurring. There's actually another protein on this surface here that's usually functioning very, very well. And this enzyme here is called adenylate cyclase. It's called, adenylate cyclase. Actually, no, I lied. This is called guanylyl cyclase. This is called guanylyl cyclase. What guanylyl cyclase is doing is it's taking and converting a molecule called GTP, and it's converting into what's called cyclic GMP. Cyclic GMP. Why is this important? Because cyclic GMP is normally bound to these special types of channels on the membrane. So it's bound to special types of channels here on the rod's cell membrane. Let's draw these channels here. Let's say here I have one channel. Okay, here I have another channel here. And then here I have two more channels here. Here's another channel. And here's one more channel. Okay, why is this cyclic GMP important? Cyclic GMP has little pockets on these channels here. It has little pockets that it can bind to. So cyclic GMP could actually bind onto this channel here. It could bind onto this channel here. And it could bind onto this channel here. And again, this channel here. When it binds onto these channels, it activates these channels here and opens up the channels to allow for specific ions to flow in. These ions are going to be sodium and calcium. If these ions are flowing into the cell, what are they doing to the inside of the cell? They're making the inside of the cell positive. If you make the inside of the cell positive, it tries to bring about a excitatory postsynaptic potential. It tries to bring the actual cell to threshold to generate what's called an EPSP. These cells, these photoreceptors, do not generate action potentials. You have to remember that. They do not generate action potentials. They generate what's called receptor potentials or graded potentials, excitatory postsynaptic potentials. But here's the problem. When light rays hits the rhodopsin, it converts 11-cis into all-trans, which frees up the opsin. The opsin activates a transducin. The transducin goes and activates this phosphodiesterase enzyme. So again, what would it do over here? It would come over here and activate this phosphodiesterase enzyme. Guess what phosphodiesterase does? It breaks down this actual cyclic GMP. It breaks down the cyclic GMP. If you break down this cyclic GMP, then cyclic GMP is no longer going to be active. If this cyclic GMP is not active, then what's it not going to be able to do then? It's not going to be able to bind onto these channels. If it can't bind onto these channels close. These channels close. And then what happens? Sodium ions and calcium ions will not move into this photoreceptor, this rod in this case. They'll actually be prevented from coming out. Coming into this actual cell, right? So let's review this real quick just to make sure it's all clear here. First thing, light rays hits the rhodopsin. Second thing, it converts 11 cis into all trans. Third thing is it takes opsin and then it frees up opsin to bind onto transducin. Transducin activates phosphodiesterase. Now, normally, in this rod, it's having GTP being converted into cyclic GMP through guanylyl cyclase. And cyclic GMP is binding onto these sodium and calcium channels on the membrane, keeping them open so that sodium and calcium can come in. But whenever the light rays are there, it activates this phosphodiesterase enzyme. And what does the phosphodiesterase do? It breaks down the cyclic GMP. If cyclic GMP is broken down, his concentration decreases. If it decreases, then he can't keep these channels open. If these channels aren't going to be open, they'll stay closed, and then the sodium ions and calcium ions can't come in. If the sodium and the calcium ions can't come in, what happens to the cell? It becomes less positive. So the cell becomes less positive. So if it becomes less positive, or we can even think about it like this, if it becomes less positive, it's actually becoming a little bit negative. So if it's less positive, it can actually become a little bit more negative. What is this called then? The cell is becoming increasingly more negative or less positive. This is called hyperpolarization. So it'll cause what's called hyperpolarization. If you guys can think about it like this, let's imagine I draw here a graph for a second. Let's say I can put here a graph. And here on the x-axis is going to be time. Here on the y-axis is going to be millivolts. Let's say here's the normal resting membrane potential. Then here's what you need to get to threshold, right? So there's your threshold potential, all right? Generally, these cyclic GMP channels are going to be keeping it kind of depolarized, right? If it's binding onto those channels, bringing the sodium and the calcium in. If it blocks it then what happens? It actually is not going to rise to threshold potential. It'll actually drop below resting membrane potential. So once this happens then, this is actually going to hyperpolarize. It goes below resting membrane potential. Okay, so that's called an IPSP. IPSP is an inhibitory postsynaptic potential. So whenever it hyperpolarizes, this produces what's called a IPSP, an inhibitory postsynaptic potential. If you pretend for a second that in red, let's say that these channels were open, so cyclic GMP was bound, then this would actually cause sodium and calcium ions to come in, making the cell more positive. It would start approaching what? It would start approaching threshold potential. If it starts approaching threshold potential, it's trying to excite the cell. This is called a excitatory postsynaptic potential. So it's trying to cause a slight depolarization. In this case, though, because light hits this, it causes it to hyperpolarize, which is called a IPSP. If that happens then, what's going to happen to the action potentials that are moving down this axon? It's going to be very, very low. Very little, if any, action potentials are going to be traveling down this axon. Then, if that's the case then, if there's very little action potentials or almost no action potentials, you know there's specialized channels down here? Specialized channels down here called these voltage-gated calcium channels. There's these voltage-gated calcium channels down here at the synaptic terminals. These calcium channels want to open so that calcium can come in. But if this cell is hyperpolarized, this is not going to activate these channels. These channels will be inhibited. If these channels are inhibited, the calcium can't really come in. And if that's the case, let's say that a tiny, tiny bit of calcium does come in. Very, very, very little calcium comes in, or almost no calcium comes in, right? You're going to release very, very little, if any, neurotransmitters. And what is this neurotransmitter that's actually getting released here? It's called glutamate. Okay? So, to pick up where we were, we said if the cyclic GMP is not bound, the channels close. If the channels close, sodium and calcium can't come in, the cell becomes hyperpolarized. That was the ninth thing. If it's hyperpolarized, there's pretty much no action potentials that are moving down the axon. If there's pretty much no action potentials moving down the axon, it's not going to activate the voltage-gated calcium channels. If pretty much no calcium is coming in, there's going to be almost very little or no release of glutamate. And if there's very, very little release of glutamate, how is that going to affect this? Let's come over here and see. Okay, let's say that this guy releases very little glutamate. So I'm going to put here glutamate. This is really weird because glutamate is generally an excitatory neurotransmitter. So you would think though, okay, it's not going to release very little glutamate. Very little glutamate is being released. This probably isn't going to activate it. Actually, it's the exact opposite. It's a very, very weird example. Very little glutamate is actually going to stimulate this bipolar neuron. Okay, it's going to stimulate this bipolar neuron. So what's happening here? It's releasing very little glutamate. And if you release very little glutamate, that's going to stimulate this actual bipolar neuron. So glutamate must be causing some type of mechanism where it's actually getting rid of cations and the inside of the cell is becoming negative. But if there's very little of that glutamate coming over here, very little cations will be leaving the cell. If very little cations will be leaving the cell will remain positive and it'll become depolarized. And in this case, it produces what's called, if it stimulates it, it produces what's called a EPSP. So this will produce a EPSP. What does that mean? It's going to generate these nice receptor potentials. Remember, it generates receptor potentials, no action potentials. The only one that generates the action potentials is really just these actual bipolar neurons, not the ganglion cells. So the receptor potentials are produced by the photoreceptors and the bipolar. The action potentials are generated by the ganglion cells, okay? So it releases very little glutamate, which produces these EPSPs, these excitatory postsynaptic potentials, which actually trigger a little bit of movement down the axon, which then release more. This is where it gets really funny. It releases a lot of glutamate. If it releases a lot of glutamate, you'd be like, oh, it's probably going to inhibit it. No, it stimulates this one. It stimulates this one, and this generates action potentials on this guy. And again, it'll release a lot of glutamate here. And if you release a lot of glutamate here, this will help to generate action potentials moving down this axon of this ganglion cell. And then if the actual action potentials are moving down this ganglion cell's axons, what does that mean then? That means that the action potentials that are going to be moving down the optic nerve is going to be increased. And if the action potentials that are moving down the optic nerve are going to be increased, that'll take it into the actual occipital lobe, specifically where the primary visual cortex is, and help us to perceive the actual image that we are seeing at that point in time. All right, so to recap that here, you're going to have light rays hitting this. Rhodopsin converts 11 cis retinal to all trans, releases the opsin. Opsin activates transducin. Transducin activates phosphodiesterase. Phosphodiesterase breaks down the cyclic GMP into the GMP, the breakdown product. Then after that, if the cyclic GMP is not present, you need cyclic GMP to keep these channels open. If these channels do not have the cyclic GMP, they stay closed. Sodium and calcium can't come in. The inside of the cell becomes increasingly more negative or less positive, and it hyperpolarizes the cell, which causes an IPSP. Very little receptor potentials are going to be moving down this axon. Very little receptor potentials. If there's very little receptor potentials, it's going to cause very little calcium to come into the axon terminals, and very little glutamate is going to get released. If very little glutamate is released onto the bipolar cells, it stimulates the bipolar cells and produces EPSPs. And then they'll generate increased receptor potentials down their axons, which will release a lot of glutamate onto the ganglion cells. If you release a lot of glutamate onto the ganglion cells, that stimulates the ganglion cells and triggers action potentials down their axons, which eventually become a part of the optic nerve. Holy crap, we did that part now. Okay. Now I want to talk about how the horizontal cells and the amacrine cells are affecting this process real quick. The horizontal cells are actually receiving, so you know whenever this glutamate is being released here, This actual guy here, so let's say here, it can release glutamate not just onto the bipolar cells, but onto the horizontal cells. And when it releases this glutamate onto the horizontal cells, it actually stimulates the horizontal cells. And then as a result, the horizontal cells release this chemical called GABA, gamma-aminobutyric acid. And then gamma-aminobutyric acid, guess what it does? It goes and inhibits this photoreceptor. You guys are probably like, wait, what the frick? I'm so confused now. Let me explain it one more time here. This guy's releasing glutamate. I'm going to put glut there, right? That stimulates this guy. This guy in response will release GABA. GABA will then inhibit this photoreceptor. The whole purpose of this is that whenever this is getting hit with light rays, you want this guy to become less sensitive to those light rays, right? You don't want him to become very, very sensitive. You want him to become a little bit more desensitized to the light rays. So what this guy is doing is, how these horizontal cells are functioning is whenever we're going from light to darkness or from dark to light, he's helping us to adapt to that. He's adapting to the changes in light, going from dark to bright light or bright light to dark light, okay? That's the function of these horizontal cells. So they're basically maintaining or modulating this actual movement, right? Or these actual photoreceptors sensitization. All right, sweet. The amacrine cells are doing very similar of that, okay? The whole function, they're still trying to sift out the function of the amacrine cells. What they do know is that the amacrine cells are involved in the connection between the bipolar neurons and the ganglion cells. And what they know is that this amacrine cell can actually release various different types of chemicals. It can release chemicals such as dopamine, it can release chemicals like acetylcholine, and it can release chemicals like GABA and glycine, many different types of chemicals. And basically what they believe that it's doing is it's inhibiting these ganglion cells. Why? So that there is very precise movement or very precise action potentials that are leaving through the ganglion cells. So again, it's modulating the actual action potentials moving through the visual pathway. The whole purpose of these horizontal cells and these amacrine cells is to modulate the actual visual pathway, to make sure that it's very precise, to make sure that we're adapting to it, to make sure that the visual field is very perfect, okay? So that's the function of these amacrine cells. Okay, so now that we talked about that, we got to talk about, okay, let's say that now you're no longer getting hit with light rays. Let's say that you actually are in dark. So now there's no light rays. If there's no light rays, then what's going to happen then? Well, then it's just going to be the exact opposite then. If there's no light rays, then won't this all trans go back to 11 cis if there's no light rays? So in the absence of light, so no light over here, right? So you get rid of this. Now you're in dark light or dim light, right? Then what's going to happen here? If there's this no light, right? If there's no light here, this is actually going to cause the all trans to go back to 11 cis. If that happens, then the opsin will go back. So now let's say it like this. Let's say it's right here. This will go from what? All trans. It'll go back to 11 cis, right? Then what will happen? Opsin will come back and rebind here. Then it'll no longer be able to activate this transducin. If it can't go over here and activate this transducin, then what happens? It's no longer going to activate the phosphodiesterase. So now it's not going to activate this transducin here. So this transducin is going to be inhibited. It can't actually activate the phosphodiesterase. So then the phosphodiesterase is going to be inhibited. If the phosphodiesterase is inhibited, he can't break down the cyclic GMP into GMP. So this process is inhibited. So then what happens to the levels of the cyclic GMP? It goes up. If cyclic GMP levels go up, then what does that mean? Oh, that means I have a lot of this here. So what would happen to all of these channels? All of these channels would open up because cyclic GMP would be bound to them. If all of these channels open up, who's going to start flowing in? Sodium and calcium. And as sodium and calcium start flowing in to this actual photoreceptors, what's going to happen then? The cell will become increasingly more positive. And as the cell becomes increasingly more positive, it's going to start depolarizing, right? But again, it doesn't generate action potentials, it generates graded potentials or receptor potentials. So again, what's coming in here? sodium ions and calcium ions. All right, so sodium ions here and calcium ions here. Coming in, and coming in, right? Then it's going to convert it from hyperpolarization. It's then going to become depolarized. And when it becomes depolarized, what's that called? It's going to generate what's called a EPSP. Now it's going to become a EPSP. If it becomes an EPSP, then what happens to the action potential, I'm sorry, not action potentials, receptor potentials moving down the axon? It's going to increase. You're going to have a lot of receptor potentials moving down. If you have a lot of receptor potentials moving down this axon, what does that mean for these calcium channels? These calcium channels are going to be super active, and calcium is going to start flowing in very, very excessively. That's going to stimulate the release of more glutamate. If I release more glutamate, what does that mean then? If I release more glutamate, I'm going to inhibit these actual bipolar neurons. If I inhibit the bipolar neurons, I'm going to cause them to generate what's called IPSPs. If they generate IPSPs, they'll have less receptor potentials moving down. If there's less receptor potentials moving down, they'll release less glutamate. If there's less glutamate, there's going to be very little action potentials moving down the axons of the ganglion cells and down the optic nerve, okay? And it's because it's in the absence of light. That should make sense. So when there's the absence of light, there's very little action potentials moving down the optic nerve. When there's a lot of light, there's going to be very many action potentials moving down the optic nerve. And again, the horizontal cells and the amacrine cells are modulating this movement through the visual pathway. Horizontal cells by releasing GABA, amacrine cells by releasing chemicals like dopamine or acetylcholine or GABA or glycine, and basically modulating the vertical transmission and the photoreceptor transmission between these guys, okay? Holy crap, I think we pretty much covered all of that part for the phototransduction. All right, now what we're gonna do in the next video, guys, is we're gonna go ahead and talk about what's called the pupillary light reflex. So we're gonna talk about what happens during dark adaptation and this light adaptation and how our pupils respond to that. And then after that, we'll talk about the visual pathway itself. Ninja Nerds, I really hope all of this made sense. I really, really do hope that you guys liked it, and I hope it helped. If it did, please hit the like button, comment down in the comment section, and please subscribe. All right, Ninja Nerds, as always, until next time.